Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. If you want to stay on the cutting edge of scientific discovery and support our work, please hit that like button and subscribe. It truly helps the Sand Science Experience grow. Today, we're embarking on a deep dive into something, well, really fundamental, how our brains age, at the molecular level. For a long time, a lot of the focus has been on things sort of breaking down, right? Like DNA damage or proteins clumping together. But this new research we're looking at today, it suggests a surprising new hallmark of aging. It kind of flips that idea. It seems to point to a problem that's happening earlier, more upstream. Not just about proteins breaking down, but maybe fundamentally about how our cells even build them in the first place.
Exactly. And when we talk about brain aging, I think many listeners probably know about this idea of declining proteostasis. And, that's not just jargon. It's this really, really delicate balance our cells have to maintain, making proteins correctly, folding them just right, and then clearing them out when they're old or damaged. Proteins are the workhorses doing almost everything in the cell. So when this balance gets messed up in aging brains, you start seeing protein damage, aggregation, things heavily linked to neurodegenerative diseases like Alzheimer's and Parkinson's. So yeah, understanding how this balance goes wrong is just absolutely critical.
Workhorses, I like that. And it sounds like if that balance is off, it's not minor. It's potentially devastating for the cell, right?
Definitely.
So this research digs into a specific puzzle in aging brains, this thing called protein transcript decoupling. Okay, so for anyone familiar with basic biology, mRNA, that's the blueprint, the instructions. The protein is the final product, the machine doing the work. Decoupling means the number of blueprints doesn't really match the number of machines you end up with. It's like having a perfect plan for a house, but I don't know, only half the walls actually get built.
Yeah.
Strange, right?
Exactly.
And to figure out why this happens in aging, the scientists use a really interesting model organism, the African turquoise killifish, Nothobranchius furzeri.
Yes, the killifish.
Yeah. Apparently, it's becoming a bit of a star in aging research. It lives a really short life naturally, and its brain ages super fast, which means researchers can see changes in months that might take decades in us humans. It's like a fast-forward button for studying brain aging.
It's an incredibly powerful model for exactly that reason.
Okay, let's unpack this then. How did they even start looking into such a complicated thing? Across different ages of the fish? What was the approach?
Well, they really went broad and deep, which is what makes this study so solid. They looked at pretty much everything involved in the protein life cycle, from the basic building blocks, the amino acids, to how proteins get modified, how well they dissolve in the cell, even where they end up physically located. And they did this comparison meticulously across young, adult, and old killifish brains. The big picture that came out of all that, a really clear pattern. Proteostasis dysfunction just gets worse and worse as the fish get older.
Right, so a general decline. But was there anything specific that jumped out within that general picture, like a certain kind of protein or maybe one pathway getting hit harder?
Yes, and that's where it gets really interesting. The core finding, the really striking thing, was this widespread reduction specifically in proteins that are rich in positively charged amino acids.
Positively charged, like basic amino acids.
Exactly, the basic ones, especially lysine and arginine. These make up lots of essential proteins, things like DNA and RNA binding proteins, really crucial stuff. And here's the kicker. The amount of these proteins was going down, but the amount of their mRNA instructions, that wasn't changing.
So that's the decoupling right there.
Precisely. A direct, clear example. The blueprints are there, but the factory isn't producing these specific parts correctly anymore.
Okay, that is specific, not just any protein, but ones heavy in lysine and arginine. So the million-dollar question, why? Why were those proteins dropping off? What did they do to figure that out?
Right. So to get to the why, they use this really powerful technique. It's called ribosome profiling, or Ribo-seq.
Ribo-seq, okay.
Yeah, so you could think of ribosomes as these tiny factories in the cell, right? They move along the mRNA blueprint, read it, and build the protein piece by piece. Ribo-seq lets scientists take a snapshot and see exactly where all those little factories are located on the mRNA strands at a given moment.
But like tracking the workers on the assembly line.
Exactly. And what they found in the older killifish brains was significant. These ribosomes, these factories, were stalling out way more often. They were pausing, sometimes even bumping into each other, like a traffic jam on the mRNA highway.
A traffic jam.
Okay.
And you said it was linked to those basic amino acids. Did the jams happen mostly when the ribosome was trying to read the code for lysine or arginine?
That's exactly it. The stalling wasn't random. It happened disproportionately on the mRNA sections, the codons. that specifically code for lysine and arginine.
Wow.
So this directly messes with the production of proteins that are absolutely critical for things like DNA repair, reading DNA correctly, maintaining chromatin structure, RNA splicing, getting RNA out of the nucleus.
Basically all the fundamental stuff that keeps a cell running and healthy.
All fundamental processes, yes. And all things we know are affected by aging. So it's not just a general slowdown. It's a specific bottleneck hitting proteins vital for cell integrity.
And if the ribosome is stalling out while it's trying to build these proteins, is the only consequence that you just get less protein? Or does something else go wrong during that stall?
Yeah, unfortunately, it's worse than just less protein. The study linked this ribosome stalling directly to increased protein insolubility.
Insolubility, meaning they don't dissolve properly, like they're clumping up.
Exactly. It strongly suggests that the proteins were actually misfolding while they were being synthesized, right there on the stalled ribosome.
So the factory isn't just slow, it's producing faulty parts right off the line.
That's a good analogy, yes.
Yeah.
Defective or improperly assembled from the get-go, which, brings up an important question people often ask about aging. Isn't the main problem just that our cells get bad at cleaning up the mess? Degrading old or damaged proteins via the proteasome, the cell's shredder.
Right. Is it a cleanup problem or a production problem?
So they tested that. They partially blocked the proteasome in adult killifish to see if it mimicked natural aging. And while doing that did cause some aging-like changes. They saw issues with lysosomes, lower mitochondrial content, things like that.
Yeah.
It did not replicate the key things they saw in normal aging, like that specific protein transcript decoupling or the big drop in basic proteins.
Okay. So inhibiting cleanup causes some problems, but not this specific set of problems.
Precisely. It suggests that while waste disposal is important, it's not the main explanation for this particular collapse in proteostasis they observed.
So what's the big takeaway then? It feels like this shifts the focus quite a bit, doesn't it? Away from just breakdown and towards, well, the building process itself.
It really does. It's a potential paradigm shift. While protein degradation definitely matters in aging, this study strongly suggests that protein synthesis specifically, problems during translation elongation, how the protein chain is built, might be a much earlier, perhaps even primary, driver of aging hallmarks.
Yeah.
It points to faulty protein building, impaired biogenesis, as a key culprit, not just the failure to take out the trash.
Okay, but what could be causing the ribosomes to stall more often? Is it a lack of raw materials, maybe? Or something wrong with the factories themselves?
So they looked into that too.
Mm-hmm.
And they did find some upstream issues. For instance, the concentrations of amino acids were different in aged brains. Arginine, one of those key basic ones, its level was cut in half.
Half, wow.
Yeah.
Yeah.
And also the overall charging of tRNAs was reduced. tRNAs are like the delivery trucks bringing the right amino acid to the ribosome factory.
So fewer delivery trucks getting loaded properly.
Sort of, yes. But while these metabolic changes, less arginine, less efficient tRNA charging, definitely contribute to the problem. They couldn't fully explain just how widespread and specific the ribosome pausing was. It suggests there are other complex factors involved, maybe issues with the ribosomes themselves or how they interact with the mRNA. It's multifaceted.
It really sounds like a potential vicious cycle, doesn't it? If the ribosomes are stalling, what happens next? That could make things even worse.
Absolutely. It looks like one. This aberrant pausing doesn't just mean slow production. Over time, it seems to lead to a reduced amount of ribosomal proteins themselves.
Wait, the proteins that make up the ribosomes?
Exactly, the very components of the factories. So fewer ribosomal proteins means fewer fully assembled. Functional ribosomes can be made overall.
So the factories start failing, which means they can't build enough parts to make new factories.
Precisely. And that scarcity of functional ribosomes then further messes up protein synthesis across the board. It's this downward spiral, a self-reinforcing collapse of the whole system, a fundamental decay of the machinery.
That's a pretty grim picture of decline. But was there anything that bucked the trend? Any surprises where things actually got better or increased?
Actually, yes. And this was genuinely counterintuitive and fascinating. Amidst all this decline, a specific subset of proteins actually showed increased abundance.
Increased. Which ones?
Components of the mitochondrial respiratory chain, for example. Proteins in complex 1, complex 2, complex 3, and complex 4, the energy powerhouses.
Huh. How could they increase if the overall ribosome machinery is declining?
Well, one hypothesis, and it's just a hypothesis at this stage, is that with fewer ribosomes overall, maybe there's less traffic on the mRNA. So for proteins that normally get started very efficiently, very high initiation rates, maybe with fewer ribosomes competing for access, These specific mRNAs get translated more efficiently, less congestion on their specific production lines, perhaps?
Okay, that is interesting. So less traffic overall might let the priority vehicles through faster?
That's one way to think about it. It highlights that aging isn't just everything declining uniformly. It's a complex remodeling of the proteome, the cell's entire protein landscape, with unexpected winners and losers.
So bringing this back from the killifish to us, to you listening, what are the big implications here for human health? We mentioned Alzheimer's and Parkinson's earlier.
Right, the implications are potentially huge. Understanding these really fundamental changes in how proteins are made in this rapidly aging fish could give us crucial new insights into human brain aging. We already know that similar issues, ribosome problems, protein misfolding, are hallmarks of diseases like Alzheimer's and Parkinson's. This work suggests that maybe problems with protein building, with translation elongation, could be a much earlier upstream trigger for other things we associate with aging, like DNA damage or epigenetic changes.
So it's not just a symptom, it might be closer to the root cause.
It could be. It shows how a specific molecular glitch, like a stutter in the protein assembly line, can have massive ripple effects on the overall health of our aging brain cells. It definitely opens up new angles for research into the very earliest stages of age-related decline.
So this deep dive really suggests that aging isn't just about stuff breaking down later in life, but maybe it's fundamentally tied to how our cells build things right from the start and how that process falters over time. What new therapeutic avenues could this open up? I mean, if we could somehow fine-tune those protein factories, support that translation process, could that help with age-related diseases? It's a really thought-provoking idea. Anyway, that's it for this deep dive. If you found this information as fascinating as we did, please make sure to like this video and subscribe to the Sun Science Experience for more exciting science breakdowns. We really appreciate your support.